question paper. So, okay, so this is our first question. Here it has been asked. So, the substrate saturation profile of an enzyme that follows Michaelis-Menten kinetics is depicted in this figure. So, what is the order of the reaction in the concentration range between 0.8 to 1.4 molar? So, from this point to this point, it has been asked the rate of the reaction. Okay, so before starting with the rate of reaction, okay, let's discuss about the Michaelis-Menten. Okay, so to start the Michaelis-Menten approach, so we all know, okay, enzyme and substrate together they form an enzyme substrate complex. Okay, and this complex releases the enzyme and substrate get converted to the product. Okay, so this is the basics of the Michaelis-Menten equation. Okay, so according to the Michaelis-Menten approach, when the rate of an enzyme catalyzed reaction is measured as varying substrate concentration. Okay, so this is the rate of the reaction, or also called as velocity of the reaction. Okay, so when the rate of the reaction or the velocity of the reaction of an enzyme, a catalyzed reaction, is measured at varying substrate concentrations. Okay, this is the varying substrate concentration. So so the rate depends on the substrate concentration. Okay, so this rate of reaction depends or is directly proportional to the substrate concentration at a relatively low concentration of the substrate. The initial velocity increases almost linearly. Okay, the reaction reaches a maximum velocity. So as you increase the substrate concentration, the rate of reaction also keeps increasing linearly until it reaches a maximum velocity at the level of. Okay, so now further the reaction, when the reaction reaches the maximum velocity, the increase in the substrate concentration does not increase any further the rate of reaction. Okay, this is a Michaelis-Menten graph. Okay, and if this is the Vmax, so the substrate concentration where Vmax is half of its maximum. Okay, and this is Vmax, and this is the half of Vmax. Okay, so at this substrate concentration is referred to as Km. Okay, or the Michaelis-Menten constant, also called as Michaelis-Menten constant, or also referred to as Km. Okay, so the lower the Km, the higher the affinity of enzyme towards the substrate. Okay, because the lower the Km will be, the more fast the reaction would be. Okay, so that tells you about the substrate and enzyme affinity towards each other. Okay, so if affinity is higher, they form complex together also very high, and the rate of reaction also increases at a higher pace. Okay, and if the Km is high, and then the affinity is also high, and then the time also takes more compared to the normal reaction. Okay, so Km denotes the affinity of enzymes or substrate towards each other. Okay, so okay, now let's talk about this graph. Okay, so according to this Michaelis-Menten equation, the reaction velocity versus the concentration shows a hyperbolic graph. Okay, so the Michaelis and Menten so when the substrate concentration was kept constant and the enzyme concentration is increased, the rate of reaction increases. But when the enzyme concentration is kept constant uh, and the substrate concentration is being increased, the rate of reaction increase to a maximal level value and then leveled off. Okay, it is caused by the saturation of all the available enzyme binding sites by substrate molecules. Okay, so talking about the enzyme kinetics, so the rate of reaction we can express in terms of either rate of disappearance of the substrate um, or rate of appearance of the product. Okay, anything can be done. Okay, so if in talking about the rate, if in a time limit, if we need to, so disappearance of the substrate in a particular time. Okay, this is the rate of reaction, or we can also say that appearance of product. Okay, you can say concentration. Okay, minus sign denotes the disappearance of the substrate and this plus sign denotes the appearance of the product. Okay, so it has been established the rate of reaction in a given time is proportional to the product of the concentration of the reactant raised to the appropriate powers. Okay, so rate is proportion to the appropriate power. Okay, or we can also say, okay, so K is a proportionality constant, also we can call it as a rate constant, and the exponent must be determined experimentally. Okay, so the exponents in the rate equation are usually some small whole number such as 1 or 2. Okay, so the value of exponents are related to the number of the molecules involved in the detailed steps that constitute the mechanism. So the overall order of the reaction is the sum of all the components. Okay, so if for example the rate of reaction is this simple like substrate to product, then rate is K if it is 1. Okay, so this is overall it is a first order reaction. If it is something like this, then okay, so it will be sum of the exponents, so it will be overall 2 order reaction. Okay, and sometimes it's the rate of reaction is 0 also when it doesn't depend on any of the uh, reactant. Okay, so if the rate of reaction does not depend on the concentration of the reactant, it is called as the zero order reaction. Okay, so that is how a rate of reaction for any particular reaction is calculated. Okay, this, so all the exponents are being calculated, but generally the value comes out as whole small whole numbers like 1 or 2 or sometimes if it doesn't depend of any reactants but depends on the enzyme concentration, then it becomes a zero order reaction. Okay, so saying the same thing, if we'll see our Michaelis-Menten reaction. Okay, so in this graph, okay, it's increasing like this. So keeping enzyme constant, the rate of reaction, okay, first it goes linearly and then reaches at a maximum level and level off. Okay, so here when it increases linearly, okay, the rate of reaction is dependent upon the substrate concentration. Okay, so at this particular point till here it follows, or you can say here till here it follows a first order kinetics. So it is a first order reaction. Okay, so when it reaches a maximum value where it does not depend on the substrate concentration anymore, only thing that can alter the rate of reaction is the enzyme concentration. So I've already told that such a reaction is called zero order, and its rate, which is constant, does not depend on the concentration of the reactant but depends on the other factors such as presence of catalyst. Okay, so that is called as the zero order reaction. So till here, till the linear uh, graph, it is following the first order reaction, and here in the next graph it is following the zero order reaction. Okay, so if we see our question, so the substrate saturation profile of an enzyme that follows Michaelis-Menten kinetics is depicted in the figure. So what is the order of the reaction? In the concentration range between 0.8 to 0.4. So it is asking between 0.8 to 0.4. That means from here to here, where it has already reached at the maximum value, and in further increasing the substrate concentration is not increasing the velocity of the reaction. The only thing that can affect this reaction is the presence of the enzyme or increase of the enzyme. Okay, so as we have already discussed, till here it will follow a first order kinetics, and from here it will follow a zero order kinetics. So the answer of the question is zero order kinetics. And this is my NPTEL reference. Thank you.